Hello friends, welcome back to my new biological video about the structure of DNA part 2. In this video, we discuss all the characteristics of the DNA. Okay, the DNA was proposed by the scientists Watson and Crick in the year 1953. But this model was created by the scientists Franklin and Euclid by experiment on the X-ray photography. But the complete DNA sequence or characteristic was proposed by the scientists Watson and Crick. Now we discuss the whole characteristic of the DNA sequence. Okay, the DNA contains 10 base pair, and each base pair are composed of sugar, nitrogenous base, nucleotide, and nucleoside. 10 base pair is equal to one complete DNA sequence. A base pair composed sugar. This sugar connected with the nitrogenous base, that is adenine, thymine, guanine, cytosine by N-glycosidic linkage. The two nitrogenous bases are interconnected by hydrogen bond, and this part is called as nucleoside. What is nucleoside? Nucleoside means when sugar is connected with nitrogenous base by N-glycosidic linkage, this is called as nucleoside. And what is nucleotide? When two nitrogenous bases are interconnected by phosphate group by phosphodiester bond, then this complete sequence is called as what? Nucleotide. The difference between nucleotide and nucleoside means when nucleoside is interconnected by phosphate group, then it creates nucleotide. When some nucleotides are interconnected one by one, a chain systematically, then a complete DNA sequence is formed. Okay, this is all about the sequence and chemical composition of the DNA. I tell you in the previous video that when adenine is connected with the N-glycosidic linkage at 3' otherwise 5' position, but the sugar where the carbon is present, this carbon interlink with the N-glycosidic linkage at first position. This was about our chemical composition or linking system. Okay, then we follow that the graphical structure of the DNA. In the graphical structure of the DNA, there are two strands are present, and these two strands are antiparallel to each other. That means one strand starting from 3' end and ends at 5' end, but another strand starting from 3' end and ends at 5' end. Okay, in a strand there are so many bases are present. In a base, adenine is equal to thymine and guanine is equal to cytosine. In 1940, the scientist Irwin Chargaff proposed that the adenine is always equal to thymine, but guanine is always equal to cytosine. Adenine links with thymine by double hydrogen bond, but guanine links with cytosine by triple hydrogen bond. In a complete DNA sequence, the length of the complete DNA sequence is 34 angstrom, and the right angle helical DNA structure is the diameter is 20 angstrom. The right angle coil structure or right angle helical coil structure of DNA is otherwise called as bDNA. A important question comes in the exam. That here a short question also arises that what is the distance or what is the distance between one DNA bases to another DNA bases. If the complete DNA sequence contains 10 base pair, then the complete DNA sequence length is 34 angstrom, then the distance between one base pair to another base pair is 3.4 angstrom, because here 10 base pair is present and the length of 10 base pair is 34 angstrom. This is all about the structure of or the characteristic of DNA. Now we know that the function of the DNA. What is the function of DNA? The main function of DNA that it stores the genetic material and it transfer from one generation to another generation, and it also control the human cell or the other living cell, and it also create some chemical system in our body. Thank you all of you to see my full video. Like and share, subscribe my channel.